Hi, good afternoon, everybody, and welcome to this latest in a series of webinars from the Francis Crick Institute. Today we're going to look at using flow cytometry to assess apoptosis. Now, those of you who've never been to these webinars before, let me introduce myself. My name is Derek Davis. I'm the STP, or Science Technology Platform Training Lead here at the Francis Crick Institute in London, and for many years I ran the core flow cytometry facility at the Crick and one of its founding institutes. And you can follow the Crick Flow Lab on Twitter, and you can also follow my training page on Twitter as well. Now this, I suppose, is slightly different to some of the other webinars that we've given, because in the past we've looked at the theory of flow cytometry or the theory of panel design and so on. This is really going to be a much more practically based webinar. We're looking at a very specific application. Now, even if you haven't done any apoptosis in the past, even if you may never want to do it in the future, what I want to try to give you is the things that you need to think about, again, for every experiment. We're always going to need to think about the cytometer. We're always going to need to think about the fluorochromes and probes that we're going to use. We also need to think about our cells and how we're going to prepare our cells to give us a good experiment. And we're also going to need to think about how we're going to analyze the data to give us the numbers that we want. And I'll try and cover most of those things during the course of the next 50 minutes or so. What I'm not going to be able to do, of course, is turn you into a flow expert or an expert in apoptosis, but hopefully I can give you some of the things that you need to think about. So hopefully you all know what flow cytometry is. It's a fluorescence-based single cell technology. And we know that we can devise quite complex experiments because the number of lasers that we have at our disposal, the number of fluorochromes and fluorescent proteins enable us to do all sorts of fancy things. So here, just simply looking at CD3 and CD8. It's not all about phenotyping, as we'll see. We can use flow cytometry to look at anything as long as we have a fluorochrome that we can attach to our cell or some aspect of our cell. And our cytometers can be either analyzers, where we get information, or they can be sorters, where we can retrieve our cells. I'll primarily be talking today about flow analysis or analysis of apoptosis. So if we take a cell, here's a cell, and I give it some sort of signal, growth signal in this case, that cell has a choice of things that it can do. If the conditions are right, that cell will proliferate, it'll start growing, and that's exactly what we want when we're growing during embryogenesis, or whether we're wound healing or regenerating. But sometimes the conditions aren't right, and a cell may choose to delete itself from the population. And it can do that in several ways. Cells can die sort of accidentally, if you like, injury. They can choose to eat themselves by a process called autophagy, or they can die by this process known as apoptosis. Or they could neither proliferate nor die and enter a process called senescence. Very important. I'm not going to talk about it at all. I'm going to concentrate today here on apoptosis. So luckily we get to talk about death in the afternoon. May not be afternoon where you are, but it's definitely afternoon here in London. So what is apoptosis? Well, you'll sometimes hear it referred to as programmed cell death. It's maybe not quite the best definition. Maybe gene-directed cell death is a better way of doing it, describing it. But this sort of leaf picture I put here and this leaf motif on the journal Apoptosis are quite appropriate, because deciduous trees lose their leaves by a process of apoptosis. The cells that are holding the leaf to the tree die, leaf falls off. It's actually a very efficient way of getting rid of something we don't want. And although apoptosis has been around for millennia, it's a very evolutionary conserved process. It's only really since the early 70s, in this classic paper from Andrew Wiley and his colleagues, that it's been studied. And of course, it's been studied in many ways. Flow cytometry is just one of those. So where do we see apoptosis? Well, we see it in lots of normal development. So amphibians will lose their tails by a process of apoptosis. We lose the webbing between our fingers during embryonic development by a process of apoptosis. In the immune system, the selection of our CD4 and CD8 cells is mediated through a process of apoptosis. T cells induce killing by inducing apoptosis in their victim cells. But of course, if we've got a process that's very widespread, it's bound to go wrong at some point. And apoptosis is quite important in things like neurodegenerative or neuroinflammatory disorders, things like Alzheimer's disease and Parkinson's disease, where we see apoptosis where we don't want it. And also in cancer, in a tumor, cells will grow out of control. They'll lose the ability to die. So apoptosis is important there as well. We don't see apoptosis where we would really like to see it. So I mentioned that apoptosis is a specific form of cell death. How does that differ from something like necrosis? Well, there are several key differences. I suppose necrosis, if you're looking at a histological section, it's very easy to see a necrotic area because there'll be the characteristic hallmarks of necrosis, a lot of inflammation, a lot of cell death. Whereas apoptosis can affect individual cells. It can affect one or two cells in a field of view, which is sort of one of the reasons why it was overlooked a lot until the early 70s. The way cells enter necrosis is different to the way cells enter apoptosis. As I said, necrosis can occur by some sort of injury to the cell, which could be mechanical, it could be chemical. Whereas with apoptosis, there's really the idea that all cells are standing on a parapet saying, I'm going to jump unless you tell me that everything is okay, and then unless I get reassurance from both the outside and the inside that it's okay for me to carry on growing. When a cell does die, it needs to be phagocytosed, and of course in most tissues, macrophages will perform that function. But with apoptotic cells, any cell can remove the apoptotic remnants of a cell. And also with necrosis, we get that inflammatory response. We'll always get a lot of infiltrating lymphocytes, whereas with apoptosis, that tends to be much more reduced, which could be an important consideration if, for example, you're looking for a tumor-killing drug. If it doesn't cause inflammatory response, if it kills cells by apoptosis, that may be a good benefit. So where is cell death important? Okay, so I mentioned 3 or 4 forms of apoptosis earlier. That was a bit of an oversimplification. And this recent publication from, which is great if you can get on the list, the Committee on Cell Death, comes up with a whole series of different ways a cell can die. So it's not just apoptosis. So I've circled the apoptotic pathways here, but you can see that only forms maybe 20% of the different ways that cells can die. So I'm going to talk principally about apoptosis here, but it's important to bear in mind that other forms of cell death are available. And again, this cartoon just shows why apoptosis can be important. Those of you who came to our DNA analysis seminar will know that looking at cell proliferation is important in flow cytometry, because one of the ways that we can look at the DNA content of cells is to measure it on a flow cytometer. And if you want to become a successful cancer cell, one thing you want to do is be able to replicate out of control. But it's not just growing out of control, you also need to be able to resist cell death. If you fell over at the first hurdle, you wouldn't be a successful cancer cell. So it's both sides of those coins. It's being able to proliferate and it's being able to avoid cell death. So, what is apoptosis? How do we define apoptosis in a cell? So there are a whole series of morphological, relatively standard morphological changes that we get in apoptosis. Cells will shrink, they start to change their shape. We get this condensation of the cytoplasm, condensation of the DNA as our DNA starts to break up, and eventually formation of these apoptotic bodies, which are the things that get phagocytosed either by macrophages or other cells. And this is sort of fairly chronological. We tend to get that changes in the cytoplasm before the nucleus, before the formation of apoptotic bodies. But at the same time, underlying those morphological changes are a whole series of biological changes or biochemical changes. Again, these are pretty chronological. So we get changes in the levels of calcium, sodium, potassium. We get the interaction of small protein molecules that are in the cytoplasm. We get changes to the mitochondria. We get changes to the actual structure of the plasma membrane. We see DNA denaturation happening very specifically, and we see this breakup of DNA. And we can put a very simple cartoon up here. Actually, it's not that simple, is it? It's quite complex, but it does show you that apoptosis is a complex process. We have these intracellular death domains. We have molecules that shuttle in and out of the mitochondria, in and out of the nucleus. We see the activation of transcription factors. We see the activation of specific molecules and enzymes. However, we're going to make it a little bit simpler. We're going to divide apoptosis in mammalian cells. I apologize again to those of you who are doing yeast or things that aren't mammalian cells. We're going to look at mammalian cells here. We can divide the apoptotic pathway into two main arms. We have what we call a death receptor pathway or extrinsic pathway. And as I mentioned, cells are almost standing on the edge, and they have to be reassured that things are okay or that they're not okay. So, the binding of Fas to Fas ligand or the binding of TNF will cause a cell to enter apoptosis, and it does it by activation of these intracellular death domains, activating this enzyme called caspase 8, which in turn activates caspase 3. And caspase 3 is sometimes referred to as the executioner of apoptosis. By the time that a cell has activated its caspase 3, it's a pretty irreversible process. It's going to die. At the same time, our cells are constantly measuring what's happening within the cell. Remember, when a cell divides, when a cell proliferates, that's probably one of the most dangerous things that it can do. So the cell is always checking for DNA damage. If there's too much DNA damage, a cell will decide to delete itself from the population rather than risk damage to the multicellular organism. But there are other things that can trigger this, levels of reactive oxygen species, for example. And so this is what we call the intrinsic pathway, and this chiefly involves the release of cytochrome C. Now, cytochrome C is normally within the mitochondrial membrane. During this process of apoptosis, it gets released. It binds with procaspase 9 to form this thing called the apoptosome, which then activates caspase 3 and initiates the whole series of apoptosis. But you'll notice these are not necessarily exclusive, because the cell could be signaled by the extrinsic pathway, but then involve the intrinsic pathway. So there are lots of different ways that a cell can end up being dead. And that's one of the things that we want to look at in flow cytometry, because we know that we're always going to add a viability dye in our flow cytometry experiments. And a viability dye normally measures the sort of end stage of death, if you like, the point when a cell is permeable, either to a DNA binding dye or a protein binding dye. We want to try and pick up cells at an earlier time point. We want to find here, really, where they've committed to cell death, but they haven't actually yet completely died. I'm sure this fellow is fine at the end of it, but this is where we're trying to pick up our cells, the physiological status before they get to what we would call a dead cell. So how can we detect apoptosis? Well, one way is to do a bit of molecular biology, run a gel, because remember, one of the things that happens during apoptosis is the DNA is chopped up into bite-sized fragments, into 180 base pair fragments, and we can detect this on a gel. We can see this characteristic laddering. We can also quantitate that a bit better by using something like single-cell gel electrophoresis, a thing called the COMET assay. Similar sort of principle. We look at the fragmented DNA because that will be able to be eluted out on an electrical field, and we can do this by image analysis or some form of image cytometry. We can also use microscopy, particularly electron microscopy, because we're really still the hallmark of apoptosis is morphology. So we can see where our cells are happy or whether they're fragmenting their DNA. Or of course we can use a flow cytometer to do that, which is why we're all here. And it's probably true to say that there are maybe 30 or 40 different methods that we can use to detect apoptosis on a flow cytometer. Now, it would be very dull if I went through all of those, but I'm going to give you a flavour of some of those. So they're based really around these five things: the way cells scatter light, the analysis of organelles, particularly the mitochondria, changes in the surface of the cell in the plasma membrane, activation of those enzymes, those caspases that we saw in the cartoon a few slides ago, and changes in the DNA content of the cells. Now, which one you use, I'll try to come back to near the end, but it's probably true to say already at this point that there is no one method that will always pick up apoptosis in all circumstances in all cells. Okay, so it may be that we might have to look at several different ways. So one very simple way to look at cell death is the way cells scatter light. As I said, we always need to add a viability dye into our flow cytometry experiment. And the way we pick up dead cells is the fact that they're permeable, in this case to a DNA binding dye, propidium iodide. So in all cases, what I've got here is an untreated population where I've just taken them straight out of the flask, and another one that I've treated with an apoptosis inducing agent. In most cases, that's staurosporin, which is a protein kinase inhibitor. But you can see that the main difference here is we can define our live cells that are being negative for propidium iodide and our dead cells that are positive. And we also know that as cells die, their forward scatter signal tends to go down. Now, if the end point of your assay is simply how many cells have died, that may be sufficient. And you will, in the literature, occasionally see people putting a marker here and saying, these are my apoptotic cells. You know, they're slightly permeable to the DNA dye, so they're not quite dead yet. I'm not sure I hold with that, but you will see it out there in the literature. But I don't tend to use this method very much to look at apoptosis. Yes, it's a good way of seeing if my cells are dead, but I don't necessarily know how they've got to be dead. So let's be a little bit more specific. Let's look at some changes in the mitochondria. So I've said mitochondria are particularly important in apoptosis, and we can look at two different things. We can look at changes in the mitochondrial membrane potential, or we can look at the release of cytochrome C. And there are several different dyes that we can use to look at either of those. So one of the ways that I like to look at apoptosis is to look at the changes in the mitochondrial membrane potential. And there are several different dyes that we have at our disposal to do that. There's the CMX dyes from molecular probes. There's a dye called LDS751, a dye called JC1, and this is its chemical name, which is why we abbreviate it to JC1, and one that I quite often use called TMRE. I suppose one thing to say about these probes is that with a functional probe, like any of these, we are stuck with the excitation and emission of that fluorochrome. Unlike antibody staining, where I can buy an antibody in virtually any fluorochrome that I can measure on my flow cytometer, with these dyes, I can't do that. I know that JC1, for example, I'm always going to be exciting with a blue laser, and I'm going to be detecting green and orange fluorescence. So that's an important thing to know, again, as it always is with any of our fluorochromes. And with these probes, we are more fixed with that. We can't necessarily change that. I'll come back to these in a second. I just wanted to mention a couple of the methods for cytochrome C analysis. One way is to use this dye called nonyl acridine orange, about which I'm not going to say anything, but it can be used to detect the release of cytochrome C. Or because cytochrome C is a protein, we can use antibody staining. And I'll show you towards the end a very elegant way of detecting apoptosis using cytochrome C antibody staining. But this is a quite common way of looking for changes in mitochondria. And most of these dyes work in the same way. In a nice healthy mitochondria, there's a proton gradient between the outside and the inside of the mitochondrial membrane. So these dyes all mostly work in the same way. If I've got a nice healthy proton gradient, I will get dye accumulating in my mitochondria. So they can't get out because they're charged. Whereas when the mitochondrial membrane potential is dissipated, I get less dye accumulating. So I see a reduction in my signal. So that's the way that most of those dyes on the previous slide worked. JC1 is a slightly different mode of action. It's what we call a metachromatic dye. So it has two fluorescence emission peaks depending on whether it's in a healthy mitochondria when it fluoresces orange, or in an unhealthy mitochondria when it fluoresces green. Now, the great thing about all of these dyes is that they're very simple. Now, I like this in a flow cytometry method. With many of these dyes, particularly if you're using suspension cell lines, you just incubate your cells for 10-15 minutes and you're ready to run them on the flow cytometer. Simpler the protocol, less things there are to go wrong. And this is the sort of result I might expect if I was using one of the CMX dyes. So in this case, I've used MitoTracker Red. So in this case, one thing that we have to remember, we always have to set up our cytometer appropriately. And remember, what we're looking for here is a reduction in fluorescence as cells enter apoptosis. So what that means is when we come to our flow cytometer, we need to make sure we set our voltages sufficiently so our live cells are pushed a little bit more to the right than we might normally put them, because we're going to be looking for a reduction in fluorescence. So normally, again, with phenotyping, I expect things to go that way. They're going to be positive. Here, I'm expecting my apoptotic cells to be negative. So here are my live cells, nice healthy mitochondria, nice bright signal. And here are my cells that have entered apoptosis. Because the mitochondrial membrane potential is dissipated, I don't get so much accumulation of the dye, and I get a reduction in the signal. Now, these cells were unfixed, so I've also been able to add a viability dye, a DNA binding dye, in this case TOPRO3. So I can separate my cells that are dead by our sort of flow cytometric terms. But you'll notice that in these two plots, there's probably not a very big difference in the percentage of cells that are dead. So if I just use my TOPRO3 fluorescence as a marker of cell death, I'd say, well, nothing has happened here. But because I've added the CMX ROS, I know that a very high percentage of my cells that are not yet permeable to TOPRO have already entered apoptosis, or at least they've already lost mitochondrial membrane potential. I wanted to put this slide in as well because it's another important practical consideration with apoptosis and cell death assays. Normally when we're using scatter to gate our cells, we would exclude these because I think they're dead and I'd be interested in this population. But of course here, the end point of our assay is cell death, so I certainly don't want to exclude those. And the only thing I do want to exclude are events that are here, down in the bottom corner. So these are events that are probably too small to be a cell. Because what I want to do in a flow cytometry experiment is make whole single cell measurements. It's particularly important in apoptosis because I think these are my live cells, and as they progress through apoptosis, they're going to go like this, but they're also going to start falling apart at the end and end up with those small apoptotic bodies, which would probably end up down here somewhere. I probably want to exclude those because I have no way of knowing whether a cell breaks up into 2, 3, 4, or 10 fragments. So to try to ensure that I'm just making measurements on single cells, I exclude the debris in the corner there. The other thing I quite often do is, again, increase my forward scatter voltage slightly more than I would do if I were doing a phenotyping experiment, if I wasn't interested in cell death, because I want to make sure that I can see the cells that appear here. And you can see here that the difference in the scatter in my treated cell. So I just want to show you another example, same sort of thing using mitochondrial membrane potential, but now using a dye called LDS751. Now, presumably you've come to these webinars because you think I have some experience in flow cytometry and I know what I'm talking about. So I'm going to tell you that those cells are live because they have a nice high LDS signal, and those cells are apoptotic because they've lost mitochondrial membrane potential and therefore have a lower signal. And you're going to believe me. You shouldn't believe me because with all experiments, we should try to check what we're saying about those cells is true. And remember, morphology is very important in apoptosis. I said I wasn't going to talk about sorting. Actually, this is a plug, if you like, for the cell sorting facility, because If this was a new dye and I wanted to convince myself that what I'm saying about these populations is true, what I can do is I can go to my cell sorter, sort a few of these cells, doesn't need to be many, a couple of hundred onto a slide, stain them with Herx, in this case, Herx 33342. So it labels the DNA of living cells. And I get what I expected to see, this lovely restful blue color, nice even staining across my nuclei. If I sort this population onto a slide, I see a very different morphology. I see the sort of morphology I expect from apoptosis. I'm getting punctate staining, marginalization of the chromatin, exactly what I'd expect from an apoptotic cell. So it is always useful to do this if you have the chance, and if you want to convince yourself of what is actually happening to your cells. Because remember, the thing that we don't get on a flow cytometer is a picture of the cell. I just wanted to show a slide using the dye JC1, which again is another quite popular way of looking for mitochondrial membrane changes. As I said, JC1 is a metachromatic dye. So what happens in active mitochondria, the dye aggregates in the mitochondria and we get an orange emission from the JC1 aggregates. When cells enter apoptosis, we don't get that mitochondrial membrane potential. The JC1 remains as monomers and we get a green fluorescence. The problem with this, of course, is it's now going to take up 2 channels in your flow cytometer. So again, this is going to be an important consideration in your experimental design. And because JC1 fluoresces green, it's not something that's very easy to use with GFP expressing cells, for example. And this is where it's always useful to have different dyes or different probes that give us the same results. So these are 3 different mitochondrial membrane probes, as we've seen, CMX-ROS, DIOC6, TMRE. These actually can all be excited by a blue laser, but this one fluoresces red, this one's green, and this one's orange. But you can see they all give pretty much the same results, if not quite the same staining pattern. So that's good because we now have some degree of flexibility in our choice of probes. So what else happens in apoptosis? Changes in the plasma membrane. And there are 2 things, or there are several things that happen to the plasma membrane, but 2 things that we're interested in. Changes in dye permeability and externalization of a particular lipid, phosphatidylserine residues. I'll start with this one actually, because this is one of the most common ways that people look for apoptosis. We can detect this externalization of our phosphatidylserine residues by binding this protein called annexin 5. And if you're used to using apoptosis, this is probably one of the assays that you've looked at. But there are other ways of doing it. As I say, changes in membrane permeability, and I'll show you some examples using a dye called YoPro1, but Herx 33342 is also differentially permeable to live, dead, and apoptotic cells. Let's look at annexin first of all. So annexin is a protein. It's not an antibody, but it's a protein. Now, because it's a protein, it means that I can buy it bound to, again, any fluorochrome that I can potentially measure on my flow cytometer. So it's very flexible. One thing about it is it's calcium dependent. The binding of annexin to phosphatidylserine is very dependent on a reasonably high level of calcium. So that means that we need to run it in a special buffer. However, it's relatively simple, so that's a good thing. However, because I have to treat my cells, I have to take my cells, I have to spin them down, I have to resuspend them in the calcium binding buffer, there's a little bit more processing there. Remember, I'm not keen on processing too much in apoptosis and cell death because, particularly when we're spinning, dead and apoptotic cells tend not to pellet as well. They're more buoyant than live cells. So we tend to lose those preferentially. So we have to be aware of that. The more spins that we do, the more chances we have of losing the cells that we're interested in. But I can get away generally with 1 wash with my annexin staining. And I'll get something that looks a little bit like this. So here are my live cells. My live cells will be negative for the phosphatidylserine. Remember, that's inside the plasma membrane. And of course, it's going to be negative for, in this case, a DNA binding dye. As my cells enter apoptosis, that phosphatidylserine gets flipped outside of the plasma membrane. So now it's being detected by the annexin 5. So we're becoming annexin 5 positive. But at this point, the cells are not permeable to the DNA binding dye, and it's only when they enter cell death that they become permeable. Now we can get this sort of assay to work on a flow cytometer or a microscope. And if I looked at the apoptotic cells, I would see that they were positive for annexin, negative for propylium iodide, but when they become dead, of course, they're positive for both. Now, one good thing about this assay is it has a sort of internal control, because my dead cells should be positive for both dyes because they're permeable. So the propylium iodide has gone in and bound to the DNA, and my annexin has also gone into the cell and bound to phosphatidylserine inside the cell as well as outside. If I see this population sitting above my live population, that generally tells me I've done something wrong. I've maybe forgotten to add my annexin, or probably more likely, forgotten to put it in a high calcium buffer. Now, the good thing about this assay is that because it's live, I can add a DNA binding dye very easily. So the time to get the results is very quick. I can get a result in this sort of assay within half an hour. But what it does mean is that I'm only looking at a snapshot of the cells that they are at this time point. So with any live cell assay, and this should be obvious, but sometimes it does get forgotten, with any live cell assay, we need to build in some sort of kinetic element. We need to do it over time. We need to start inducing apoptosis here and then look at our cells at different time points. Because this is an example from one of our users, and they knew that morphologically these cells were becoming apoptotic, but if they looked at the percentage of cells in this annexin 5 positive region, they didn't see much of a change over time. But remember, this is a dynamic process. Cells will start here, then they will externalize their phosphatidylserine and become annexin positive, and then they will die and they'll become permeable to propylium iodide. So they're going to move. They're going to move throughout the course of this experiment. So if we plot what we should have done, the amount of cells that are apoptotic or dead, then we see the expected rise that we saw by microscopy. So one question that we get asked with annexin is, these cells are obviously dead. They're brightly positive for propylium iodide. But what about these things here? Death isn't an all or nothing procedure. Well, it is. But remember, what we're looking at here is permeability. And cells don't suddenly become impermeable and then immediately permeable. There will be a gradation. But here, this is a Jurkat cell line. So dead cells are hanging around in the suspension. So it's equally possible that maybe these cells were cells that died a long time ago. They became permeable and now they've started to break up and lose DNA. So they're becoming less positive. How can I separate those? And this is where one of the dyes that I mentioned that is based on dye permeability comes in, a dye called YoPro-1. Now, YoPro-1 is a DNA binding dye. But we know that cells are very self-protective, aren't they? They have all sorts of membrane pumps on their surface, and those membrane pumps act on molecules of different shapes and sizes. So there are different pumps for dyes like propylium iodide and dyes like YoPro-1. And it was found that YoPro-1 went into cells very quickly after they'd entered apoptosis. Presumably the membrane pumps that normally would kick this dye out break down very early in the apoptotic process. So here we have our usual untreated and treated plots. Here are my live cells, which are again negative for YoPro, and in this case we've used DAPI as a viability dye. Here are my cells that are entered early apoptosis. The membrane pumps that normally would keep YoPro-1 out of the cell are broken down, so YoPro-1 enters the cell, binds to the DNA. Then cells start to become permeable, they die. So cells will become positive for both YoPro and DNA. But you'll notice there's a sort of parabola effect here, because both YoPro and DAPI are DNA binding dyes. So the more of both I get into the cells, the more quenching of the fluorescence of the YoPro I get by the amount of DAPI. And then as cells enter necrosis, they're starting to break up their DNA. So I get a reduction in the signal from both of those DNA binding dyes. So I can have what I like to call the parabola of cell death. So it gives us a little bit more information. Potentially it allows us to define early permeable cells, late permeable cells, and then cells that are breaking up their DNA. And if that's something you want to get out of your experiment, this may be something that's worth investigating. Remember, we can also detect enzyme activation, and particularly those caspase enzymes that I talked about earlier. Caspase 3, 8, and 9 are particularly important. And of course, they're proteins, so we can have an antibody against them. That's easy, and we can get an antibody that's directed against the whole molecule, or we can get an antibody directed against the active form of the molecule. And again, that could be useful information. I could do dual antibody staining in different colors and see what proportion of my caspase 3 load in the cell was active or inactive. But because it's an enzyme, we can also use the enzymatic activity of the caspase to detect its presence. And that's this thing here that's a kit called 5-Lux, which is from a company called Oncoimmunin. And this is based around actually one of the earliest types of flow cytometry experiment. If you add a non-fluorescent substrate to a cell, that can be cleaved by specific enzymes within the cell to yield a fluorescent product. And that's the basis of this assay. So here are my live cells that don't have any active caspase. Here are my apoptotic cells. So they have active, in this case, caspase 3, which is acting on the non-fluorescent substrate, the 5-Lux substrate, to yield a fluorescent product. Now, the advantage of this sort of technique, because it's enzymatic, you're going to get an amplification. The more enzyme you have in there, the more substrate you have in there, and the longer you incubate for. And because it's a live cell assay, we can also exclude our dead cells on the basis of DNA staining. And you can see that here, the dead cells don't have any caspase 3 activity, as we would expect. As I said, we can also detect caspase activity by an enzyme. From a functional point of view, this is quite a dull slide because it's immunophenotyping, isn't it? It's looking at my untreated cell binding caspase 3, and here's my treated cell in blue. So most of the cells have active caspase activity. But I put this in here because quite often your reviewers will ask you to have checked whether your cells that you're calling apoptotic are expressing caspase 3. So we can also detect apoptosis by looking at the DNA. Remember though, the DNA changes tend to be fairly late in the apoptotic pathway. And we can do this detection by two different methods, or several different methods, but two common ones. One of them is looking for DNA fragmentation by this thing called the sub-G1 method, which again, if you've done any apoptosis analysis, is probably one of the methods that you're likely to have come across. It's a very common method of looking for what people refer to as apoptosis, but as I'll show you in a second, I don't like to describe it like that. Or we can look for broken strands of DNA by this technique called tunnel, which I'll also describe in a moment. But we can also use, again, immunofluorescence staining to look at things like gamma H2AX, which is normally activated in broken strands in DNA, or PARP staining. So why is sub-G1 popular? It's popular like annexin because it's very simple. Basically, we just fix our cells in ethanol and we stain our cells with a DNA binding dye. In this case, I've used propylium iodide. But the principle is that as cells die, they start to fragment their DNA. So if I fix my cells in ethanol, remember ethanol is a coagulating fixative. If I fix my cells in ethanol, those small fragments, that low molecular weight DNA, will be washed out of my cell when I prepare my samples. And what that will mean is when I stain those cells with a DNA binding dye, the cells that had fragmented DNA will now have less DNA, they'll have a lower signal. So just a couple of things to be cautious about here. When we do our gating, remember we always try to exclude doublets in DNA analysis, and we do that generally on the basis of the height and the area of our DNA signal. Single cells will fall up the diagonal, and normally I would put my region approximately here. But because I'm looking for cells that have fragmented DNA, I have to make sure that my gate here is extended almost to the origin. Again, not quite the origin, for the same reason that I excluded those events that were near the axis in scatter, because I don't know what these are. These could be apoptotic bodies that have come from multiple cells. So I exclude the things that are in the bottom corner. And then I look at my DNA histogram, and you can see that the difference between my untreated and treated is that I've got a population of cells here in the sub-G1 region, which is where the name comes from. And you will see in the literature people putting a marker around here and saying, I've got X% apoptotic cells. I'm not sure I entirely agree with that, because I think what you can say is that you've got at least X% of cells that show DNA fragmentation. But remember, apoptosis isn't the only process where we get DNA fragmentation. It happens in necrosis as well. And also, if we think about a cell that was in G1 when it entered apoptosis, let's say it fragments 20% of its DNA. So my signal would be reduced by 20%. So that cell would have been here originally, but would end up here after we've stained it. Great, I would see it in the sub-G1 region. But what's happened if my cell's in G2 when it died and entered apoptosis? If it loses 20% of its DNA, now it would end up somewhere in here. So I'd be underestimating the amount of DNA fragmentation if I just use this figure. However, it is a very quick and easy way of seeing if something is happening, certainly if we're seeing DNA fragmentation. So again, here's a time course where we treated our cells with an apoptosis inducing agent, and we see this population here in sub-G1 increasing over time. But what this would tell me is that I should try to assess apoptosis by another technique as well as this one. And one other way of looking at DNA fragmentation is to use a strand break labeling technique, which we call tunnel, which stands for terminal deoxynucleotidal transferase mediated DUTP nick end labeling. Again, this is why it's abbreviated. We don't want to be saying that all the time. But it uses an enzyme, TDT, to bind a fluorochrome labeled nucleotide to a broken strand in DNA. Now, the difference between this and the sub-G1 method, remember in the sub-G1 method, we fixed our cells in ethanol. Here, we fix our cells in an aldehyde, formaldehyde generally. And formaldehyde is a cross-linking fixative. So those little fragments of DNA are now no longer washed out of the cell, they're fixed in place. So it means that I can visualize those strand breaks, but I can also get an idea of the cell cycle status of the cells with broken strands. So I can simply get a percentage of cells that show strand breaks here, but because I've combined the nucleotide incorporation with propidium staining to look at DNA content this time, I can be relatively specific about whether my cells were in S, whether my cells were in G1, S, or in G2, where they became apoptotic. So again, it gives me maybe a little bit more information that I might get from some other techniques. To be honest, we don't use tunnel as much as we did a few years ago. It is very useful in immunohistochemistry. Some of you might be aware of using it on tissue sections, because again, it's picking up cells with strand breaks, but with tissue sections, we also get that positional information about where cells that have entered apoptosis were in relation to their neighbors. So what I've talked to you about so far is really single probe methods. Okay, most of the time, yes, I've added a DNA binding dye, but it's not particularly high dimensional. And of course, we know that flow cytometry, one of the big advantages of flow cytometry is that it allows us to integrate methods, to multiplex, to look at lots of different fluorochromes together. So in the last 10 minutes or so, I just wanted to give you some examples of that. So here's an example where we've used 4 different dyes. TMRE, tetramethyl rhodamine ethyl ester, which is a dye that allows us to measure mitochondrial membrane potential. Annexin, which looks at the externalization of phosphatidylserine residues. 7-AAD, which I've used as a viability dye in this experiment. And I've used Herx 33342, which, if you recall, is a dye that will go into live cells and quantitate the DNA. So if I look at this plot, this is my typical annexin viability dye plot. Annexin against 7-AD in this case. And these are my cells that I would be calling apoptosis if I were just looking at annexin. However, the great thing in this experiment is I've got 2 other dyes. So if I gate on my negative cells, so these are cells I would normally be calling live, if I look at the TMRE against Herx signal, I can see that in this population that looks like it's live by annexin staining, actually half of my cells have already lost mitochondrial membrane potential. And it's difficult to see on this plot, but actually there's more cells losing mitochondrial membrane potential from G1 than the other phases of the cycle. And if I look at these cells that were annexin positive, actually all of those have reduced TMRE staining. So that's saying to me that by the time cells are annexin positive, they've already lost their mitochondrial membrane potential. So because again, this is a live cell assay, I can do it over time. So I can plot the percentage of cells that are positive by TMRE versus annexin, so the orange and green lines here. So that shows me that cells are entering apoptosis via the mitochondrial route before they externalize their phosphatidylserine residue. And we don't start to see any sub G1 till the cells start to externalize their phosphatidylserine residue. So we can use the multiplexing to help us get better or more information. So here's an example again, using caspase 3. Remember, caspase 3 is looking for, we're looking for caspase 3 activation, sorry, by staining it with an antibody. So what we've done here is we've fixed our cells in formaldehyde, then we've permeabilized them, because remember formaldehyde isn't a very good permeabilizing reagent, and then we've stained with both the caspase 3 and propidium iodide. So I can see the cells that are caspase 3 positive, and I can see here that there doesn't seem to be any cell cycle phase specificity, because here's my cells in G1, S, and G2. Remember I said we can look for cytochrome C release. Now by flow cytometry, this is actually quite difficult, isn't it? Because although cytochrome C is a protein and therefore we can have an antibody directed against it, what we're seeing in apoptosis is simply a movement of the cytochrome C. In a healthy cell, it's in the mitochondria. In an apoptotic cell, it's outside the mitochondria. So if I were to stain a healthy cell, I would pick up all of the cytochrome C that was in the mitochondria. Whereas if I were to stain an apoptotic cell, I would still pick up the cytochrome C, but now it's no longer in the mitochondria. So how can we think about how we could do this? Well, one way to think about it is, remember, when we permeabilize our cells, we don't always have to use a fix and permeabilizing step. We can sometimes just use a permeabilization step. That's what we're going to do here. We're going to permeabilize our cells at this point. So in my healthy cell, the mitochondria, and therefore the cytochrome C, will stay within the cell because they're big enough to be retained in the cell. Whereas with my apoptotic cell, because cytochrome C is quite a small protein, it will be washed out through the holes that I've punched in the membrane by my detergent. So the detergent we use is a detergent called digitonin. So what we do here is we permeabilize our cells with digitonin for 10 minutes or so, then we fix our cells with ethanol and do our antibody staining. So what we then see is that our untreated cells, all of our cells are cytochrome C positive, whereas in our treated cells, a good proportion of our cells have lost cytochrome C staining. And the additional advantage here is because I've used permeabilization and ethanol fixation, small molecular weight DNA has also been removed from the cells, and I see a sub G1 population as well. So it's a sort of bit of a trick, if you like, to look at translocation in a system where we can't look at our cells. But sometimes we do want to look at our cells, don't we? And of course, microscopy is very useful there, but so is imaging flow cytometry. So we can do the similar experiment here using imaging flow cytometry. Similar sort of approach. If we're going to stain our cytochrome C and we're going to stain our mitochondria with a mitochondrial dye. So we use this dye called MitoTracker Deep Red to label our mitochondria, and we use an FITC labeled antibody to label our cytochrome C. Now in a healthy cell, we're going to see a co-localization of the signal, aren't we? Because the mitochondria still contain that cytochrome C. Whereas within an apoptotic cell, although we're going to see both signals, they're not going to be co-localized. And this is something that we can use imaging flow cytometry for, because we can derive what we call a co-localization score. So if we have high co-localization, as we will in a healthy cell, we get a big score, a big number. When we get low co-localization, that number decreases. So what does that look like in practice? So again, here's my untreated cells, treated cells. Nice cytochrome C, it's marginalized, it's sort of punctate, it's showing that it's in the mitochondria. You can just about see the mitochondria here in the red. When I merge them, we get a good co-localization, so a high co-localization score. When I treat my cells enter apoptosis, the cytochrome C leaves the mitochondria, so now we don't see them co-localized. We see this more punctate, diffuse staining, or diffuse staining throughout the cell, and our co-localization score has gone down. So it shows that we don't have to use a traditional or conventional flow cytometer to look at apoptosis. We can also use an imaging flow cytometer. So what should we remember when we're doing an apoptotic experiment? Well, we should remember that within an individual cell, once the cell has decided to enter apoptosis, it goes through apoptosis very quickly, generally sort of 15 or 20 minutes. And there are some great time-lapse videos on YouTube that show cells going through apoptosis. But what it does mean as well is that we need to think about time as a parameter, because a cell may not enter apoptosis immediately after it interacts with an inducing agent. It may have to be in a particular phase of the cell cycle. It may have to have a particular concentration of the inducing agent and so on. So we should always remember to look at things over time, and that time frame may depend on the inducer of apoptosis. We also shouldn't assume that signs of apoptosis will be seen. As we've said, pretty much all the methods I've mentioned are looking at a very specific part of the apoptotic pathway, and cells don't have to go through that pathway. Some cells, for example, will never externalize phosphatidylserine. Keratinocytes tend not to go through that form of apoptosis. Cells don't always become caspase 8 positive, for example, after they've been induced apoptosis from the external environment. So, which is why we should do more than one method. If we're looking at adherent cells, we should remember, if we're doing it on cultured cells, to collect the supernatant. Our dead and apoptotic cells will lift off the flask much more easily. So if we throw those away, we're almost selecting for our live cells. So we have to bear that in mind as a consideration. And also, because we don't get the picture of the cell in flow, we should use what we find in conjunction with other methods, particularly morphology, whichever way you want to do that, light microscopy, imaging, flow cytometry, or EM. So I said there isn't one method that we'd always choose. So, which method would you choose? Well, of course, my answer to every question in flow cytometry is it depends. And it's going to depend on the cell system you're using. Methods that look at membrane permeability may be less appropriate for adherent cells, because simply getting those cells off the flask, you may induce some membrane damage. Not always, but again, it's a consideration. We should think about how we've induced cell death, particularly if we know how we've induced cell death. So one way of inducing cell death is to induce cell damage into the DNA. So it maybe doesn't make sense to use sub-G1 or tunnel as a readout, because we'll be looking at the cause rather than the effect of that inducer. It's always going to depend on what else we want to look at. If we're just looking at cultured cell lines, that probably won't be much of a problem. But if we've got a heterogeneous population and we want to use phenotypic markers to define particular subsets, then it's going to be more tricky, isn't it, to put these into a panel. It may also be dependent on your cytometer, the number of lasers, the number of detection channels that you have in there. And obviously it also depends on cost. Cost is a big important thing to everybody, and the simplicity of the methods. I like simple methods that are involving live cells, but if I'm using live cells, I have less flexibility in terms of storage, for example. And as always, it depends on the expertise available. And then finally, just to summarize the methods that we've looked at, there are methods that look at apoptosis using either live cells, which could be viability, mitochondrial potential, looking at the membrane, or looking at the activation of those enzymes, or ones that we can use on fixed cells, again, looking at the proteins, caspases, or the fragmentation of the DNA. And which one you use, or which ones you use, will really be up to you. And if anybody's got any questions, I'll try and answer those in a minute, but here's the last slide for today. My usual reminder that if you go to the Crick What's On page, that will give you an update on the upcoming webinars, and we have them planned, I think, all the way through June. You can follow me on Twitter, as I said earlier, or you can also follow the RMS, the Royal Microscopical Society, or Flow Cytometry UK, both of which put on courses and have lots of resources available. Or you can email me either on the stp-training or derek.davis@crick.ac.uk. So if you think of something next week, please feel free to email me. I'll be quite happy to answer any questions. You will get a link to a feedback questionnaire after this webinar. It's very short. Please take a couple of minutes to fill it in because it does help us decide on what other webinars might be useful for you out there. And all the webinars are recorded and will be put up onto our YouTube site. This is the playlist here. I'll let you know when this one goes up. There's normally a couple of days lag before we get it up. But everything that we've done, there'll be at least one version up there for you to look at. Okay, so I'll stop there and I'll take any questions that we've got. So we do have one question that says, can I fix PI? It's just a very good question because I suppose it depends on what you're looking for. Okay, so obviously we stain cells with PI for two different reasons mostly. One to look at viability and one to look at the DNA content of the cells. And if I'm looking at DNA content, I do want to fix my cells. I fix my cells and then I do my PI staining, and that will quantitate the amount of DNA in the cells. However, if I'm doing viability, I can't fix the cells because no matter whether I fix them in ethanol or formaldehyde, that propylium is not fixed in place. So it will leach out again. So if I do need to fix my cells, I can't use a DNA binding dye. I would normally use one of those amine reactive dyes or live dead fixable dyes. And another question that says, do you think that phosphatidylserine exposure could be a reversible event? Well, I think that the further you go up towards the beginning, if you like, of the induction of apoptosis, the more evidence there is that at that point things are reversible. So as I said, by the time cells get to be caspase 3 positive, it's thought that pretty much that's it. They've decided that they're going to die. But there is some evidence that certainly at the mitochondrial membrane potential stage, that can be a reversible event, and possibly even at the phosphatidylserine event. I think this is going to depend on both cell type and the nature of the inducer of apoptosis, which is one of the reasons why I always suggest looking at a time course and different methods of apoptosis. Okay, so and then another one for looking at radiation-induced damage. How useful is annexin 5? I think that's supposed to be. Well, annexin 5 radiation-induced damage is probably likely to be nuclear DNA damage. So because annexin is looking at a plasma membrane change, I would still say that that's useful in that situation. Again, my caveat would be I wouldn't just do annexin staining in those cases. I would look at other areas of the apoptotic cascade. So again, looking at activation of caspase and looking at some form of mitochondrial change. So another question, can I use TMRE, annexin FITC and DRAX7 together? Yes, you can do that because of the nature of the excitation and the emission of those dyes. So with TMRE, I can excite with either a blue laser or a yellow laser, and it emits in the orange region of the spectrum. FITC will be blue laser excited and green, and DRAX7 we would use a red laser and far red emission. So that combination actually works quite well, and it's one that I use quite a lot because I normally don't just do annexin or TMRE. It's very easy to combine those two together. It's a very simple protocol. Okay, so I think that's the end of the questions. Say if you do think of anything, please feel free to email me. Please keep an eye on your email because I will send all attendees the link to the recording of this session, and I'll also send you this link here, which is the YouTube playlist. So thank you all once again for coming, and I hope to see you soon on another webinar. So enjoy the rest of your day.